Hello and welcome to the live session of week 8 of NPTEL course Experimental Biochemistry. I am Surasree. I am a Prime Minister's Research Fellow of IIT Kharagpur. So today in our live session we will discuss about enzymes. How enzymes work as catalysts, what are the different types of enzymes. If we plot the kinetics of enzyme or the catalysis of enzyme, how will the plots look like? What's the mechanism of action of enzyme? We will discuss one particular enzyme on the enzyme mechanism of action of one particular enzyme, and also we will discuss about inhibitors. So how does inhibitors inhibit enzyme action? Now let's begin. So enzymes are protein catalysts. So enzymes, they are protein catalysts. So not like to I just want to clarify it in the beginning, that is enzymes are usually mostly proteins, but there are also other types of macromolecules, like for example RNA, which can work as an enzyme. So enzymes can also be RNA, and enzymes can also, so they are known as ribozymes, and enzymes mostly they are proteins. So enzymes, they perform the action as a catalyst. So we know in any reaction, in any chemical reaction, what is the role of a catalyst? It speeds up or accelerates the rate of reaction, but it does not participate in the reaction itself, and after the reaction it remains unchanged. So the same thing applies for an enzyme also. So they are biological catalysts. So enzymes also accelerate the reaction rate by reducing activation energy of the reaction. Now what is an activation energy? So here we can see this graph. This is a graph of the free energy change, that is G or the Gibbs energy with the progress of reaction. And if we plot the free energy change of a reaction, we will see that the substrate, they have a higher energy than the products. That is why this reaction, when substrate is converted to product, there is a free energy change, right? So when substrate is converted to product, there is a change in free energy, and this free energy change, which is delta G, this delta G is usually negative for spontaneous reactions. So for reactions which occur spontaneously in our nature, for these reactions, the delta G value, which is the free energy change, is negative when substrate is converted to product. So therefore, when the energy change is negative, then we can understand that substrate has a higher energy and product has a lower energy. Here also we can see the same thing, that the reactants or the substrates have a higher energy than the products. Now there is also another barrier that these substrates has to overcome to be converted to products. This is the barrier that I am talking about. So this energy barrier, so the substrates from this position, the substrates has to reach this position, which is actually called the transition state. So the substrates, if they want to be converted to the products, so the substrates have a tendency to be converted to product. Why? Because the product has a lower energy. So the substrate, if it has to be converted to the product, then it has to cross a energy barrier, and this energy barrier, so when it goes up this energy barrier, it exists in a state which is called a transition state. It exists in a state which is called a transition state. Now we can see that this energy barrier is very high for an uncatalyzed reaction. So which means that the reaction, when it is happening without the presence of any enzyme or any catalyst, right? So here, since it is a biological reaction we are talking about, so we are talking about enzymes. Now this reaction is occur, if this reaction has to occur in absence of the enzyme, then the barrier, the barrier of this activation energy is much higher. So this energy is called the activation energy, the energy which is required for the substrate to be converted to its transition state, that is called the activation energy. Now this barrier is much higher when there is no enzyme, but we can see that when there is presence of enzyme, that means catalyzed, then this is the barrier, or this one, right? So then the barrier becomes much lower, or the energy transformation, that barrier is much lesser when there is presence of enzyme. So the activation energy is reduced. So that is the function of the enzymes. The enzymes accelerate the rate of reaction by reducing the activation energy. So we can see when there is presence of enzyme, we can see that this is how the curve is looking like, right? So this is how the curve is looking like. So therefore the substrates still have to reach its activation state or transition state. The substrates still have to reach its transition state, but the it requires less energy than when there was no enzyme. So since there is less energy barrier, so therefore what happens that this substrate can reach the products much earlier or much faster. So that is how the enzymes accelerate the rate of reaction by reducing the activation energy. So I hope you understand that this is the principal concept or the basic concept of enzymes, that is they reduce the activation energy. So this red arrow that I have drawn here, this is actually the activation energy when there is enzyme. Now let's look at what are the different types of enzymes. Now enzymes, so before going to the classes of enzymes, first let's talk about the two types of enzymes which are known as simple enzyme and complex enzyme. So what is simple and what is complex enzyme? So simple enzyme is enzyme when this is a whole protein, but a complex enzyme is an enzyme where there is a small organic molecule associated with it, which is known which can be called prosthetic group or a coenzyme. Now what is a prosthetic group, what is a coenzyme? First let's talk about it. So first term that we will discuss here is apoenzyme. Apoenzyme is the enzyme where there is no such cofactor or coenzyme or prosthetic group is associated with it. So apoenzyme is only the protein enzyme, the whole protein, where there is no such molecule which is attached to it. That is called the apoenzyme. Now the holoenzyme is equal to apoenzyme plus prosthetic group or coenzyme. So there is something associated with the apoenzyme, then this enzyme is called a holoenzyme. And remember that this holoenzyme is actually the functional enzyme. So there are some enzymes which cannot function without its cofactors or prosthetic group or coenzyme. So then that in that part is called the apoenzyme, but the holoenzyme, it is a functional enzyme which is associated with prosthetic group or coenzyme. Now what is a prosthetic group? A prosthetic group is nothing but a small organic molecule which is bound to the apoenzyme by covalent bond. That is a prosthetic group. And what is a coenzyme? The coenzyme is when a small organic molecule is bound by non-covalent interactions. So in case of a prosthetic group is named as a molecule which is bound by covalent interaction, and coenzyme is a molecule which is bound by non-covalent interactions to the enzyme. Now these terms you must remember while we are discussing about enzymes. Now let's talk about the classes of enzymes. So there are several classes of enzymes and they are denoted as EC, enzyme class. Now this is EC 1, 2, 3, 4, 5 and 6. So all the enzymes, they are classified under these 6 enzyme classes. So the first class is oxidoreductase. So what is the function of an oxidoreductase? It acts to add or remove hydrogen atoms. So it's a reductase, that is why. So it adds hydrogen atom or removes hydrogen atom. Now the next one, second class of enzyme is transferase. So transferase, by the name you understand that it transfers functional groups between donor and acceptor molecules. For example, kinases. Kinases is one type of transferases. So what kinases does is that they transfer phosphate groups from ATP to other molecules. So that is the role of kinase. So it's a transferase. Third is the hydrolase. Hydrolase adds water across a bond and thus hydrolyzes the bond. So its function is hydrolyzing. Next is the lyases. Lyase is an enzyme type which adds water, ammonia or carbon dioxide across double bonds or remove these elements to produce double bonds. So that's the function of lyases. Next, coming to isomerases. Isomerases perform isomerization reaction. So they perform many kinds of isomerization reaction. For example, L to D isomerization, mutase reactions. So what mutase does? It shifts the chemical groups from one bond to other. And similar, there are different types of isomerization reaction that these isomerases can perform. Next, coming to the ligases. Ligases join two chemical groups. So their function is joining two chemical groups with the energy from ATP. So they hydrolyze ATP, and with the energy that evolves from ATP hydrolysis, they can perform this joining function. So these are the several classes of enzymes, and all the enzymes are distributed in these classes and they have specific numbers. So all enzymes can actually be, so under these classes there are many subclasses also, and all the enzymes can be distributed in these classes and subclasses, and then they are named or they are given a particular number, and by that number we can identify the enzyme. So that is the classification of the enzymes. Now coming to the catalysis activity of the enzymes. Now while we are discussing the catalysis activity, we will first discuss the Michaelis Menten kinetics. So what does the enzyme do? The enzyme converts the substrate to product, right? And in between, when it binds to the when it binds to the substrate, then it forms the enzyme substrate complex, which is called the ES. So if we look at this scheme, we will see that this E is standing for enzyme and S stands for substrate. So the enzyme and the substrate, they combine to form the enzyme substrate complex. So this is an intermediate which is very important for the kinetics measurement of Michaelis Menten or enzymes. So this is the enzyme substrate complex, the ES. Now we can see that there is a reversible arrow here. So the enzyme can combine with the substrate with a rate constant of small k1, and the ES, the enzyme substrate complex, can also break down. So that is why it is reversible. It can also break down to form enzyme and the substrate. So to release enzyme and substrate, and this breakdown of enzyme substrate complex or ES has a rate constant of k minus 1. So the formation of ES has this rate constant k1, and formation or breakdown of ES has this rate constant k minus 1. Now this ES, the enzyme substrate complex, now what it will do is that it will further break down to release the product and the enzyme. So the enzyme will not change, so it will be released unchanged, and there will be, the substrate will be converted to the product. Now this rate, this is given this rate constant k2. So where this enzyme substrate complex breaks down to form the product and the enzyme, this has this rate constant k2. Now in any reaction, the slowest step of the reaction is known as the rate determining step, because it actually on this step the rate of the enzyme reaction depends. So that is why here this k2 is the rate determining step, or the formation of the product from the enzyme substrate complex. That is the slowest step, and that is why this is the rate determining step. Now to determine the kinetics or the velocity of the enzyme reaction and how it is progressing with the substrate concentration, how does it vary with the substrate concentration. So to discuss that, the first thing that we have to assume here is this steady state assumption. What is a steady state assumption? So basically it is actually considering a steady state when the formation of ES and the breakdown of ES has the same rate. So basically with respect to time, the rate of change, so the rate of change of ES is 0. So basically with respect to time, there is no change in ES concentration or enzyme substrate concentration. Why? Because the rate of formation of ES and rate of breakdown of ES is equal. So that is an assumption. With this assumption, we begin the determination of the velocity of the enzyme reaction. So with this assumption, we will begin it. Now I will come to this curve later on, but first let's discuss the kinetics, how we derive the Michaelis Menten equation. So I told you that so this is the reaction when enzyme and substrate, they combine to form ES, and then ES is converted to enzyme and product. Now what is the rate of formation of ES? The ES form, so the rate of formation of ES can be written as this: K1 into E into S. This is the rate of formation of ES. Now let's talk about the breakdown of ES. So the ES can break down to give enzyme or product, or it can also break down to give enzyme and substrate, right? So since this is a reversible reaction, So the breakdown of ES will have two components. One is when the K1 into ES, K minus 1 into ES. So when enzyme substrate complex breaks down to give enzyme and substrate, so K minus 1 into ES, and K2 into ES. So K2 into ES is when the enzyme substrate complex breaks down to give rise to enzyme and product. Now from the steady state assumption, we know that the rate of formation of ES will be equal to the rate of breakdown of ES. So that is what we write. So we equate these two. So we write that K1 into E into S equals to K minus 1 into ES plus K2 into ES. So that is the assumption that we do from the steady state assumption. Now the enzyme concentration can be written as this. So enzyme concentration equal to total enzyme concentration minus the enzyme substrate concentration. So enzyme concentration, that is the free enzyme concentration at any point, is basically the difference between total enzyme and the enzyme substrate. So the difference between total enzyme and how much enzyme has already formed a complex with the substrate. So that is what we write here. In place of E, we substitute it with E total minus ES into S, and the rest of the equation remains unchanged. Now what we do is we multiply this part and we get K1 into ET into S equal to, and we take this K1 into ES into S to this right hand side, and we get this, that K minus 1 plus K2 plus K1 into S into enzyme substrate concentration ES. Now what we do is, now in the next step we divide both sides by K1. So ET into S becomes equal to ES into K minus 1 plus K2 by K1 plus S. Now this K minus 1 plus K2 by K1, this is actually known as the Michaelis Menten constant, or Km. So this is actually the Km, or the Michaelis Menten constant. So in place of this K minus 1 plus K2 by K1, we write Km. So in the next step what we do is we take this here in the left hand side, ET into S equals to ES into Km plus S, and we take this ET into S in the denominator of this right hand side, and then we get ES equal to, we take this Km plus S as the denominator in the left hand side. So we get ES equal to ET into S divided by Km plus S. So the enzyme substrate complex equals to the enzyme total concentration into the substrate concentration divided by the Michaelis Menten constant plus the substrate concentration. Now there is another thing that we have to see here, that is, in this step when the product is formed, so the product formation rate actually depends on this enzyme substrate concentration. So this dP dt, or the rate of change of product formation with respect to time, we can write as dP dt equals to K2 into ES. Right, so we can write this dP dt equals to K2 into ES. So from this what we do is here we have this ES. So in the next step what we do is we multiply with K2 on both sides. So we get K2 into ES equals to K2 into ET plus into S divided by Km plus S. Here this K2 into ES is nothing but the initial velocity of the reaction, because the K2 is the rate determining step, so that gives us the velocity, initial velocity of the reaction given as V0, and K2 into ET, gives us the Vmax. So when this K2 is multiplied with the total enzyme concentration, it gives us the maximum velocity. So now we substitute this, we substitute here in this particular equation with V0 and Vmax, and we get this Michaelis Menten equation, which states that V0 equal to Vmax into S divided by Km plus S. So that is how we derive the Michaelis Menten equation. And so here what we see is that this V0 or the initial velocity is dependent upon the substrate concentration. Now if we plot this equation, how will it look like? So it looks like this. This particular graph is the Michaelis Menten plot. So where we have taken the substrate concentration in the x-axis and the V0 in the y-axis. And here we see that when there is low substrate concentration, so the rate of enzyme or the velocity of this enzyme, or the V0, the velocity increases. But when this substrate concentration becomes greater, so at greater substrate concentration, what happens? This curve, this becomes hyperbolic. So it does not follow any linear pattern anymore. So the linear pattern, the linear part of this graph is only when there is less substrate concentration. So in this Michaelis Menten curve, we can see that the substrate concentration, this is a curve of substrate concentration with respect to the velocity, and here we see that when there is very less substrate concentration, then this V0 is linearly proportional to the substrate concentration, but then with increase in substrate concentration, the velocity does not increase linearly, but after a certain point it starts to form a plateau. So why does this happen? So when the substrate concentration is less, there is more enzyme than with respect to the substrate, right? So the enzymes are present, which have these active sites, or the sites where the catalysis or the enzyme reaction takes place, right? So the enzyme active sites are not saturated in this portion. So in this part we can say that since there is less substrate concentration, so the enzyme active sites are not saturated. But so that is why when we add more and more substrate, the enzyme velocity is increasing, because more and more enzyme active sites are being occupied by the substrate. But as we increase the substrate concentration more further, then what happens? Then the enzyme active sites are now all saturated. So that is why there is a plateau that forms because of the saturation of the active site of the enzyme. So this Michaelis Menten curve actually states that when we increase the substrate concentration, initially the velocity of the enzyme will increase, but after a certain point, even with increase in substrate concentration, the velocity will not increase. So therefore we can say that this enzyme has reached its maximum velocity. So this is when it is known as the Vmax. So that is called the maximum velocity or the Vmax. So here this is the point which we call as Vmax. So when the substrate concentration is much more than the enzyme concentration, then this velocity becomes equal to the Vmax. And there is another term which is the Km. So the Km, the Michaelis Menten constant, that also we can determine from this graph. That is, So when V0 equals to half of the Vmax, then the substrate concentration becomes equal to Km. So how does this occur? So basically, when V0 equal to half Vmax, what we will do is we will put it in the Michaelis Menten equation. So the Michaelis Menten equation is V0 equal to Km into S divided, Vmax into S divided by Km plus S. So here when V0 equal to half Vmax, we put half Vmax equal to Vmax into S divided by Km plus S. So we can cancel Vmax from both sides, and we will get Km plus S by 2 equal to S, right? Or we will get Km equals to S. So when we can say that when V0 equals to half Vmax, that is when the velocity of the enzyme is half of that maximum velocity, then the substrate concentration is actually gives us the value of Km, or the Michaelis Menten constant. And also from this graph we see that when there is low substrate concentration, the velocity is proportional to substrate concentration, but at high substrate concentration, when this saturation occurs, then the velocity is independent of the substrate concentration as the graph starts to form a plateau. So that is all about Michaelis Menten equation, and this is the derivation of the Michaelis Menten equation. Now next, coming to one important characteristic of the enzyme, which is the turnover number and Km. So turnover number is what? It is basically the number of substrate molecules that is turned over or catalyzed in the enzyme per second. So this is the maximum number of moles of substrate that can be converted to product per mole of catalytic site per second. That gives us the turnover number. Now this turnover number, or kcat, we denote it as kcat, it is a direct measure of the conversion of substrate to product. So kcat is a direct measure of the conversion of substrate to product. Now kcat is also, we can also determine kcat as the rate constant for the rate determining step. So here in this particular Michaelis Menten kinetics, we were talking about this K2 as the rate determining constant, right? Because this enzyme substrate complex, when it breaks down to form product and the enzyme, that is actually the rate determining step or the slowest step. So the rate constant of the slowest step is actually the kcat value. So here the kcat for Michaelis Menten is actually the K2, and kcat can also be written as this: kcat equals to Vmax divided by total enzyme concentration. Now what is Km? Km we have already discussed, that is the Michaelis Menten constant. Now Km is an indicator of the affinity of the enzyme for the substrate. So low Km indicates high affinity. So basically when the Km is low, then we can say that this enzyme has high affinity for the substrate. When the Km is high, we can say that the enzyme has low affinity for the substrate. And when we divide kcat by km, so this ratio of kcat by km, it is an important ratio for measurement of the catalytic efficiency of the enzyme, kcat by km. Right, so high kcat by km indicates an efficient enzyme. So when there is this, the value of this ratio, this kcat by km, when it is high, then we can say that the enzyme is more efficient. That means if this ratio is high, either this kcat value is high or the km is less, and we know that when km is less, the enzyme has high affinity. So therefore this kcat by km is a measure of the catalytic efficiency of the enzyme. Now we have talked about this Michaelis Menten curve, Michaelis Menten graph, how it looks like. It looks like a hyperbola when we plot the substrate concentration with the initial velocity of V0. So if I go back again, when we plot the substrate concentration with the V0, it gives us this hyperbolic curve. Now from a hyperbolic curve, it is difficult to get certain values like the slope, and when we linearize the curve, if we can linearize the curve, it becomes easier for us to determine certain values and also extrapolate the curve, because a hyperbolic curve cannot be extrapolated. So there comes several plots which have actually linearized the classical Michaelis Menten curve, and one of them is Lineweaverburk plot. So that is a double reciprocal plot. So why is it a double reciprocal? So in Michaelis Menten, the curve was V0 versus S. Now in Lineweaverburk, what Lineweaverburk had done is that he had done reciprocal on both the axes. That is, he had plotted 1 by V0 with respect to 1 by S. So that is why it's a double reciprocal on both axes. And when this 1 by S is plotted with respect to 1 by V0, we get this kind of a linear curve. Now from this linear curve, we can get several information. That is, so this is the Michaelis Menten equation. V0 equals to Vmax S into Km plus S. Vmax S divided by Km plus S. And when we make double reciprocal, what we get is 1 by V0 equals to Km by Vmax into S divided by 1 plus Vmax. Plus 1 by V. So it is 1 by V0 equals to Km by Vmax into S plus 1 by Vmax. So we can say this 1 by Vmax is actually the y-intercept, because this is the it will we can so this is a linear plot. Here our y is 1 by V0, our x is 1 by S, so our m or the slope is Km by Vmax, and our y-intercept is 1 by Vmax. So this is the y-intercept. So this gives us the value of 1 by Vmax. So from this curve, the calculation of Vmax becomes very easy. It can only be calculated from this y-intercept, and also the Km value can be calculated easily. That is by so this 1 by Km is actually the x-intercept. So this is the y-intercept and this is the x-intercept. So the x-intercept gives us the value of minus 1 by Km. So minus because it is in the negative x-axis. So that is minus 1 by Km, and the slope of the curve, which can be determined easily, that is the Km by, that gives us the value of Km by Vmax. So we can see that from the Lineweaverburk plot, it becomes very easy to determine these values. Now Eadie Hofstee also has given us linear plot of Michaelis Menten equation, and they have plotted this V0 with versus V0 by substrate concentration. So enzyme velocity by velocity with respect to velocity by substrate concentration. That also gives us a linear graph, and here also the slope becomes minus Km, and this point is the Vmax by Km, and this upper part point is the Vmax. So the y-intercept is the Vmax and the x-intercept is the Vmax by Km. So here in Lineweaverburk plot, this x-intercept was in the negative axis, but here the in Eadie Hofstee plot, the x-intercept, and the total graph is in the positive axis. So any doubts so far? Okay, we will move on to the next part of our discussion, where we will discuss about the active site of the enzyme. Now the active site is basically the site of catalysis of this enzyme. Now we can see this is a three-dimensional structure of an enzyme, and this part, this small portion, is actually the active site. So this is the site where the substrate will bind. Now active site has residues, the specific amino acid residues, which are important because of their several factors or their several characteristics which we will discuss. And the active site may also contain some cofactors. Now what are the properties of the amino acid residues of this active site? These residues which are present in the active site, they are important because of their charge, the pKa value, their hydrophobicity, flexibility and reactivity. Now why are these characteristics necessary for the active site residues? Because the active site is the site where the substrate will bind. Now this active site is complementary to the geometric shape of the substrate. So we know that enzymes are specific for substrates. Each enzyme is specific for a substrate, and this substrate specificity of this enzyme actually originates from the fact that the substrate can fit in the active site of the enzyme and can fit properly. And this fitting means that there has to be the shape of the substrate has to be complementary to the shape of the active site of the enzyme. So this geometric complementarity is very important. Along with that, the amino acid residues which line the active site, they interact with the substrate in several ways, and these interaction, this can be charge based interaction, so electrostatic interaction. So active site residues, they contain these charged residues, they are charged residues. Their pKa is also very important because the buffer in which the enzyme will be functional and the substrate will remain stable. Then the hydrophobicity, the flexibility. So this enzyme, actually there is a conformational change that occurs in the enzyme. We will see it later on when we will discuss the how does the substrate binds and the several models that explain the substrate binding to the active site. And then we will talk about this flexibility, that the enzyme has to be flexible to accommodate the substrate. So the flexibility of these amino acids and the reactivity of these amino acids is very important at the active site of the enzyme. So this actually shows when the substrate has occupied the active site and there is a conformational change that we can see. So here in this particular enzyme, this enzyme exists in a particular conformation like this, but we can see that these two arms as if they have closed to contain the substrate now, or to bind to the substrate now. So this kind of conformational change also exists in enzymes. Now this binding of the substrate to this active site, it can be explained by several models. So initially, or in the very beginning, scientists have assumed the enzyme and substrate binding as a lock and key model. So that means just how a lock just how a key fits into a particular lock, and any key will not fit into any lock, right? So the key which is specific for the lock will only fit into the specific lock. right? So there is some specificity in the lock and key. Similarly, there is specificity in the enzyme and the substrate. So only the substrate, only a particular specific substrate will fit into the enzyme, and the enzyme is so designed that is it can fit the substrate. So here we can see this red one is the substrate here. This is the substrate and this is the enzyme. So we can see that this substrate, the shape of the substrate, this shape of this substrate is complementary to the shape of the enzyme, and this substrate has fit into the enzyme as if it has gone into the correct lock. Right? So this was a very initial assumption that and also it explains if there is a substrate with a different shape, it will not be able to fit into the enzyme. Right? So this was a very initial assumption. Later on, this induced fit model was actually discussed, and the induced fit model states that there is there is a flexibility in the structure of the enzyme. So the flexibility is given by these the amino acid residues which are present in the enzyme. So what we can see that the enzyme here, this model actually states that the enzyme here is not an exact fit of the substrate, right? So it does not exist in a state where it is absolutely complementary to the substrate shape, but when substrate is added, there is a conformational change that occurs in this enzyme. We can see now that it will be able to fit the substrate. So this kind of conformational change upon binding to the substrate is actually discussed in this induced fit model. So it is different from the lock and key model in this way that the enzyme and the substrate, they are not exactly absolutely complementary to each other. So that is they are not like lock and key, but the enzyme is a dynamic molecule and it has some flexibility, so it can change its shape as required for the substrate binding. There is a third model also which is called the conformational selection model. So it states that there is a conformational change that occurs to the enzyme prior to ligand binding. So even before ligand can bind or the substrate can bind, there is a conformational change that occurs. So see, here is the substrate, but in this enzyme population, this enzyme is changing from one conformation to another, right? And substrate binding, actually the substrate here seems to be selecting the right conformation for its binding. So that is why it is called conformational selection. So these three models try to explain how the substrate can bind to the active site of the enzyme. Now in this respect, we will discuss the mechanism of action of this enzyme called RNase A. So it is an RNase or ribonuclease derived from bovine pancreas. So it is bovine pancreatic RNase A. So RNase or ribonuclease, right? Ribonuclease means it can cleave off RNA or ribonucleic acid. Right? Now the mechanism of action of ribonucleic acid, to discuss that, we have to first check what are the residues which are present at the active site of the enzyme where the reaction is going to take place. So this is actually gives us the shape of the active site of the ribonuclease A, and here two important residues are present. One is this histidine 12, another is this histidine 119. So these two residues are present at the active site. Histidine 12 and histidine 119. These are the residues of the enzyme which are present at the active site. Now these histidine residues, they alternatively act as base or an acid. So how does that happen? So this molecule, this is an RNA molecule that we can see here, right? This is an RNA molecule, ribonucleic acid. So what happens first is that this histidine 12, this acts as a base. So we know that bases, they donate electrons. So when this RNA molecule comes into the active site of this enzyme RNase A, this histidine, the lone pair on the nitrogen of this histidine, it donates its electron to this hydrogen atom, right? And thus it accepts this proton. So this proton is comes to the histidine 12, and therefore this OH bond, this OH bond is broken. So if I zoom in a little bit, this electron pair is donated to the proton, and therefore the proton becomes associated with this histidine 12, and therefore this OH bond, this bond breaks, and now this O actually forms bond with the phosphate group, right? And this phosphate, from this phosphate group, there is this phosphodiester linkage or phosphoester linkage here. This one is one phosphoester linkage. This phosphoester linkage breaks, and then it interacts with this hydrogen of the histidine 119 in the active site. So then what happens? So this actually what it does is that this bond is broken. So here we can see that this bond is now broken, and this phosphoester linkage that joins the nucleotide number 1 to the next nucleotide, so that this phosphoester linkage that joins two nucleotides, this phosphoester linkage breaks. So this phosphoester linkage breaks, and this has now accepted this proton, and this phosphoester linkage has broken. So that is the role of this enzyme, to break the phosphoester linkage. Now how will it release so this particular base, this particular nucleotide is released from the active site, and there is addition of water. So now what water does is that now in this second step, here we can see that this particular histidine now acts as an acid. So in the first step it was acting as a base. In the second step, now it acts as an acid, right? And it donates this proton. So it donates this proton to this particular bond, and therefore the second histidine, that is a histidine 119, it also accepts the one hydrogen atom from the water molecule. So it accepts a hydrogen atom from the water molecule, and so it breaks this bond with this particular nucleotide. So this the attachment of this histidine with this nucleotide is broken with the help of this water molecule, right? So because of that, what we see here is that now this nucleotide also becomes free. So in the first step, the second nucleotide was freed, and in the second step, the first nucleotide is also now freed. So now what we have seen here is in this RNase A active site, these histidine residues are the important players for this catalysis. So in the first step, the histidine 12 acts as a base, and it accepts this hydrogen atom, right? So because of which this phosphoester linkage breaks, and in the second step, this histidine molecule now acts as an acid. And then what happens is that this second nucleotide is also released from the active site. So now the RNA chain is finally broken. So that is how the RNase A can catalyze the hydrolysis of RNA. That is why it is called hydrolysis, because there is this water molecule which helps in the release of the RNA molecule from the active site, and also there is breakdown of this phosphoester linkage between the two nucleotides. Now in the next part, we will talk about the inhibition of enzyme. How enzymes can be inhibited with the help of several inhibitors. So enzyme inhibition can be of two types: reversible inhibition or irreversible inhibition. Now reversible basically means that the inhibitor reversibly bind and dissociate from the enzyme. So therefore, when the inhibitor is removed, on complete removal of this inhibitor, the enzyme activity is recovered. And this reversible inhibition is possible when the binding of the inhibitor to the enzyme is usually non-covalent in nature. So that is why this is a reversible binding, and so when the inhibitor dissociates from the enzyme can regain its activity. So there are three types of reversible inhibition that we will discuss in this session. That is the competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. That is, these are the three types of reversible inhibition. And what is irreversible inhibition? So when the inhibitor irreversibly associates with the enzyme, so it does not dissociate from the enzyme, and that is why it is usually covalent in nature. So the interaction between the inhibitor and the enzyme is usually covalent in nature. So even if we remove the inhibitors, the enzyme will not regain its activity. Now we will discuss the three types of reversible inhibition. The first is the competitive inhibition. So this schematic actually shows what type of inhibition it is. So here this is when a substrate binds to the active site of the enzyme. So this is a normal substrate, it binds to the enzyme. But here in case of competitive inhibition, so the competitive inhibitor is a type of inhibitor which binds to the active site. So there is a competition, that is why it is called a competitive inhibition. So there is a competition between the substrate and the inhibitor. So this one is the substrate and this one is the inhibitor. So there is a competition between the substrate and the inhibitor, because the inhibitor comes and sits at the active site of the enzyme, right? So there is a competition that is occurring between the substrate and the inhibitor. So now if there is an inhibitor, so how will the velocity of the enzyme be changed with respect to substrate concentration? So here we plot the Michaelis Menten curve in presence of a competitive inhibitor, and what we see is that this is the graph of a normal enzyme when there is no inhibitor. But when there is competitive inhibitor present, so the Km value becomes different. So there is a increase in the Km value. So we will also see it here. Again, we will discuss it in terms of the Lineweaverburk plots. But So the Km value here will increase in case of a competitive inhibitor. So why is this Km value increasing? So this Km actually denotes the affinity of the enzyme for the substrate, right? So when there is an inhibitor, since the inhibitor is binding to the active site of the substrate, so obviously the substrate is now having less affinity for the enzyme. The enzyme is having now less affinity for the substrate, because the inhibitor is binding to the active site of the enzyme. So therefore the enzyme now has less affinity for the substrate. So now the Km value increases, for the Km value of the enzyme increases in presence of competitive inhibitor. We can see that the Km is basically half Vmax, so right? So if this is approximately half, we can see the Km for the enzyme without inhibitor is less than the Km for the enzyme with inhibitor. So this Km with inhibitor is higher than the Km without the inhibitor, right? So that happens in competitive inhibition. Now we will now discuss non-competitive inhibition, and we will again come back to this curve. But what is non-competitive inhibition? So it basically means that there is no competition. So what is non-competitive inhibition? It is something where there is no competition between the substrate and the inhibitor. So naturally we can say that the inhibitor does not bind in the active site of the enzyme, but the inhibitor binds to some other site of the enzyme. So we can see it here in this schematic that this is the substrate and this is the inhibitor. Now the inhibitor binds to the to some other site in the enzyme, which is called the allosteric site. So the enzyme, in the allosteric site of the enzyme, the inhibitor binds, and what it does is that it changes the shape of the active site. So it basically induces a conformational change of the active site of the enzyme, right? Because of which now the substrate, so here you will see that this substrate has a complementary shape to the enzyme active site, and this is the allosteric site. So this is active site and this is allosteric site. Now the substrate can bind to this active site and the inhibitor binds to the allosteric site. So now once the inhibitor binds to the allosteric site, there is a change in conformation of the active site, and therefore the substrate will not be able to bind to the enzyme anymore. So therefore there will be no reaction, or inhibition of the reaction will take place by this non-competitive mode. Now when there is non-competitive inhibition, so what changes can we expect in the Km and the Vmax? So the inhibitor binds to the enzyme and does not allow the substrate to bind to it, but the affinity of the enzyme for the substrate does not change. So therefore in non-competitive inhibitor we will see that the Km value will not change. So here the Km value for the enzyme without inhibitor as well as the enzyme with non-competitive inhibitor is same, because the affinity of the enzyme for the substrate does not change, but the Vmax value changes. So you can see that the Vmax value of the enzyme without inhibitor was much higher, but when there is non-competitive inhibitor, the Vmax of the reaction decreases much, right? So there is a reduction in the Vmax value when there is non-competitive inhibitor present. Now next, coming to the uncompetitive inhibition. So uncompetitive, the speciality of this uncompetitive inhibition is that the uncompetitive inhibitor binds to the enzyme substrate complex. Okay, so it binds to the complex of the enzyme and substrate, when there is a complex formation of the enzyme and substrate. And then when this inhibitor binds to this enzyme substrate complex, it forms this enzyme substrate inhibitor complex or ESI complex, which is a dead end complex. So there will be no reaction after the inhibitor binds to this enzyme substrate complex, and it will not allow the substrate to be converted to product, right? Now in this case the substrate is binding to the enzyme, but the inhibitor is binding to the ES complex. So now what changes are we expecting in this kind of uncompetitive inhibition? Will there be a change in Vmax and Km? So we can assume that since the inhibitor is binding to the enzyme substrate complex, and this inhibitor binding is occurring after this formation of enzyme substrate complex, we can assume that the Km value will not be affected because the enzyme is binding to the substrate. But the Km value is affected here because the substrate which binds to the enzyme, it cannot be converted to product, right? So the substrate, the affinity of the substrate therefore reduces. So that is why in case of uncompetitive inhibition, we will see that the Km value will reduce and the Vmax will also reduce in this case. Because why? Because of this inhibition, there will be less reaction occurring, right? When the inhibitor will bind to the ES complex, so there will be less or reduction in Vmax. So now if we plot this inhibition in inhibition kinetics in this Lineweaverburk plot, we will see for competitive inhibition, we have already discussed this, that the Km value will increase because the affinity for the substrate will decrease. So Km will increase, but the Vmax will be unaffected. So we can see that 1 by Vmax remains same, but the Km changes. So the Km is the x-intercept. The Km changes, but the y-intercept, it remains constant. So the 1 by Vmax or the Vmax value remains constant. In case of uncompetitive inhibition, we can see that there is change in both Km as well as Vmax. So this y-intercept also changes, the x-intercept also changes. So Km is reduced as well as Vmax is also reduced in case of uncompetitive inhibition. And in case of non-competitive inhibition, the Km value is unaffected because the inhibitor is binding to an allosteric site, not the active site. So the Km value is unaffected. So what we can see here is that the minus 1 by Km or the x-intercept is same for when there is inhibitor and when there is no inhibitor, but the Vmax is reduced. So 1 by Vmax is reduced for non-competitive or 1 by Vmax is increased for non-competitive inhibition. So when there is inhibitor, the 1 by Vmax has increased. So therefore we can say that Vmax is reduced, right? So that is how we can plot the kinetics of the enzyme when there are presence of these several types of inhibitors. And these all, again I'm telling you that these all are reversible inhibition. There is no such irreversible inhibition occurring in this part. Irreversible inhibition occurs when the inhibitor binds to the enzyme covalently and destroys the enzyme. So the enzyme will not be functional anymore. Any doubts so far? Okay. All right. Then we will move on to another type of inhibition which occurs in metabolic pathways usually. So the metabolic pathways, if you have studied glycolysis or if you have studied Krebs cycle, you know that there are several reactions that compose or that consist one metabolic pathway. and several reactions are taking place, and there are several enzymes which are involved. So in these pathways there is something which is called a feedback inhibition, which is a very common type of inhibition of enzymes in such metabolic reactions. So feedback inhibition is nothing but when there are multiple enzymes, we can see, and this is the threonine. The threonine is the initial reactant, and the enzyme 1 is threonine deaminase. So threonine is converted to isoleucine. Threonine is an amino acid which is converted to isoleucine, right? This is the overall reaction, threonine converted to isoleucine. Now there are multiple steps in it. So the first enzyme is actually called threonine deaminase. So it causes deamination of threonine, right? So from threonine, by the help of these several enzymes 1, 2, 3, 4 and 5, there are 5 enzymes in this pathway. So the threonine is in the sequentially converted to become isoleucine. Now the first enzyme is threonine deaminase, and this isoleucine is an inhibitor of this threonine deaminase enzyme. So this isoleucine actually binds to this allosteric site of the enzyme which is threonine deaminase. So isoleucine can bind to the threonine deaminase allosteric site. So this isoleucine serves as the inhibitor. So it binds to the allosteric site and does not allow binding of the substrate to the enzyme, right? So that is how the enzyme, the threonine deaminase enzyme, can be inhibited. And if the first enzyme is inhibited, then obviously the rest of the pathway will also not function, because the rest of the pathway will depend on this product A, which is the product produced by threonine deaminase, right? So threonine is first converted to A, then converted to B, then to C, then D, and then finally to isoleucine. These A, B, C, D, I am not elaborating what are these products, but you can always go and check in detail. So here the concept is that this final product of this pathway is an inhibitor of the first enzyme, right? So that is called feedback inhibition. Why does this kind of inhibition exist? To check, to control the level of isoleucine production. So if the isoleucine production has occurred much more than required, then what will happen? This isoleucine will go and bind to the allosteric site of the threonine deaminase enzyme, and it will further inhibit the process, right? So that is how the feedback inhibition takes place. Any doubts so far? Okay. So that was all about today's discussion. So we discussed about the enzyme catalysis and how does enzyme work as a catalyst. So it reduces the activation energy of the transition state, and there are several classes of these enzymes. Now the enzyme kinetics can be followed by Michaelis Menten kinetics, where we plot the substrate concentration with respect to the initial velocity, and it gives us this hyperbolic curve where the reaction actually is V0 equals to Vmax into S divided by Km plus S. So this is the equation, V0 equals to Vmax into S divided by Km plus S. Here we assume that there is a steady state, which basically means that the formation of ES and the breakdown of ES, that is the enzyme substrate complex, those two rates are same. So that gives us a steady state assumption, and from the steady state assumption we derive this equation that V0 equals to Vmax S into Km divided by Km plus S, which we plot here, and we get this kind of Michaelis Menten curve. Now this Michaelis Menten curve can be converted to linear curves in this way. this is a double reciprocal plot where we plot 1 by V0 with respect to 1 by S, or we can plot it by V0 with respect to V0 by S. And from these linear curves, it is easier for us to determine the values of the Vmax, Km. So the y-intercept gives us the value of 1 by Vmax, and the x-intercept gives us the value of minus 1 by Km. And we also have discussed about the turnover number, the kcat and the km. So this kcat by km, we have discussed that it is an indicator of the catalytic efficiency of the enzyme. High kcat by km value indicates a much more efficient enzyme. We have also discussed about this active site of the enzyme, how it determines binding of the substrate. Now the complementarity, or there is a complementarity that is present between the substrate and the enzyme active site. So this complementarity can be geometric and also can be electrostatic. So the residues that line the active site of the enzyme, those residues they have electrostatic interaction with the substrate. Now the substrate binding to the active site can be explained by these three models: lock and key, induced fit model, and conformational selection model. And we have also discussed the mechanism of action of RNase A, how the two histidine residues play key roles in RNA breakdown or RNA cleavage by RNase A. And then we have discussed about the different types of reversible inhibition: competitive inhibition, where the inhibitor sits at the active site and competes with the substrate; non-competitive inhibition, where the inhibitor binds to the allosteric site of the enzyme; and uncompetitive inhibition, where the inhibitor binds to the enzyme substrate complex. And we have also discussed about the changes that occur when there is competitive, uncompetitive, or non-competitive inhibition, the changes in Vmax and Km values, and the feedback inhibition process, when the final product of any metabolic pathway becomes or inhibits the first enzyme of this metabolic pathway to check the level of production or level of breakdown of any particular product. Now let's go to our question answer session. Please let me know your answers in the chat box. Yes, the correct answer is B, active site. Next question. Yes, Amardeep, do you want to answer? Yes, correct. So the correct answer is B, hyperbolic. So that is how the shape of the Michaelis Menten curve will look like when we plot the velocity in x-axis with substrate concentration in x-axis, velocity in y-axis, and substrate concentration in x-axis. Next question. What does it mean if delta G is negative? No, your answer is not correct, Madi. So delta G is negative, it means that it is a spontaneous reaction. So the reaction will be favored towards product, because from S, when we go from S to P, this energy change is negative, right? So therefore product will be favored. So therefore the correct answer is product. The reaction equilibrium will favor product, the formation of the product. Is that okay? Okay. Next question. Okay, so the quantities that can be measured: substrate concentration, you can measure how much substrate you are providing, how much product you are getting, that also you can measure, and you can also measure the total enzyme concentration, because that is also known to you. So the correct answer is substrate, product, and the total enzyme. Those quantities are measurable. Next question. Okay, yes, D is the correct answer, because we talked about this, that enzyme acts as a catalyst, so it does not change. So the enzyme will be, this X will actually be the enzyme itself. Next question. Okay. No. Aarti, your answer is not correct. To measure enzyme kinetics, we vary the substrate concentration. So remember the Michaelis Menten curve. The substrate concentration is varied with respect to the velocity, and we get this kind of a Michaelis Menten curve, right? So substrate concentration is varied to measure the enzyme kinetics, that is to measure the Vmax and Km values. Next question. Okay, Aarti's answer is perfect. So the substrate specificity is actually determined by two points: geometry and electrostatics of the substrate and the enzyme. So the geometry of the substrate and enzyme has to be also fitting each other, so they have to be, their shape has to fit each other, and also they also need to be electrostatically interacting. So the correct answer is A and B. Next question. Okay, Amardeep, you are correct. It is Km is the concentration of the substrate when V0 equals to half Vmax. That means when the velocity of the enzyme is half of that maximum velocity, then the substrate concentration becomes equal to the Km. So which is why we can get the Km value from the x-axis of the Michaelis Menten curve, or in the Lineweaverburk plot also. The substrate concentration we can, or the Km value we can get from the x-axis. So that is why it is not the enzyme, the concentration of the enzyme required to reach maximum velocity. It is the concentration of the substrate required to reach half of the maximum velocity. So correct answer is C. Is that okay, Aarti? Alright. Okay, next question. Amardeep, do you want to answer? Okay, so the correct answer is in Lineweaverburk plot. So not only it linearizes Michaelis Menten equation, but obviously since it becomes linear, it is easy to extrapolate a straight line. So therefore we can extrapolate certain values, and also the important parameters like the Km and Vmax, those can be determined from the intercepts of the two axes, right? From x-axis we can determine, x-axis intercept we can determine the value of -1 by Km, and from y-axis intercept we can determine the value of 1 by Vmax, right? So the correct answers here are A, B and D, all three of them. Okay, next question. This is just, I told you just now. Yes. So the y-intercept gives the value of 1 by Vmax. Correct. Okay, next question. So think about it intuitively, that is what will happen if we increase the enzyme concentration, or if we increase the substrate concentration in case of competitive inhibition, or if we decrease the substrate concentration or enzyme concentration. So where we can obtain maximum velocity, in which of the conditions? Okay, so in case of competitive inhibition, so the substrate is competing with the inhibitor, right? There is a competition between the substrate and the inhibitor. So in which case we can increase the velocity, or in which case we can get maximum velocity of the enzyme? So if we increase the substrate concentration, yes, if we increase the substrate concentration, then only our substrate, so more and more substrates will compete with the inhibitor, and then so the substrate will outcompete the inhibitors, and therefore the enzyme can reach its maximum velocity, right? So that is why if we increase the concentration of the substrate, the velocity will also increase in case of competitive inhibition. Next question. Is that okay, Aarti, the previous question? All right. Okay. Here the right answer is allosteric site. Correct. The next question. Okay. The correct answer is competitive inhibition, right? Next question. Check your answers again, both of you. Absorbance is one factor, surely. Absorbance is something that we monitor, that is sure. But with respect to what? Wavelength is not the correct answer. So absorbance we monitor with respect to time, because we are monitoring enzyme activity. So with respect to time, what is the change that we can observe in the absorbance? That will actually, yes. So the correct answer will be D. So time will be in the x-axis and absorbance will be in the y-axis, right? So that is how we will be able to monitor the activity of the enzyme. And the last question for today. That actually this I did not discuss, but if you have gone through the lectures of week 8, you might know. If you have gone through the practical demonstration. Yes, actually the correct answer is oxidation of phenolic compounds in apple by the enzyme orthodiphenol oxidase. So that is the correct answer. Okay, so that's all for today. If you have any doubts, please let me know. And in the next Tuesday we will again meet for a week 9 session. And if you have any doubts, please let me know. You can email me or you can check my other videos from my YouTube channel. So thank you so much for joining. We will meet again in the next week.